Neurophysiology deals with the mechanism of generating an impulse, which is an electrical signal or an action potential. So, when we talk about neurophysiology, we oftentimes refer to what is called the membrane potential. And this is a form of stored energy, actually. When we look at the membrane potential, this is the difference in charge between the inside and the outside of a cell. And in the cell that we're talking about here is the neuron, and the part of the cell that we're talking about is the axon. So this difference in membrane potential can be measured using a little voltmeter, and it's measured in millivolts. When we look at the difference, we could look at a section of the axon, and we could say that the inside is negatively charged compared to the outside. So this is at rest. We have what is referred to as the resting potential. So this is when a neuron is not sending an impulse. And the resting potential is measured at a negative 70 millivolts. Now, occasionally something happens and this changes. It actually reverses. And then we get a positive charge on the inside and a negative charge on the outside. So this is what is referred to as an action potential, and this is an impulse. And so the action potential is measured at 30 millivolts. Now, this action potential is a brief reversal of the charge, so that if we look at this, we could see that before and after that action potential, there is a reestablishment of the resting potential. So I'm just going to write here is my action potential. And then right here and right here at the end would be my resting potential. So this is a change in the charge, which is a type of electrical energy. So if we look at what changes this charge, it has to do with the movement of ions. And so when we look at the resting potential, we find that there is a higher concentration of sodium ions outside of the neuron than inside the neuron. So let's look at the ions. So at rest, sodium ions are more concentrated outside the axon than inside. So if we were to open a channel, the sodium ions would diffuse. Passive diffusion doesn't take energy. It just goes from a higher concentration to a lower concentration. So sodium ions will want to move in to reestablish the equilibrium. And then we also find that the potassium ions, so this is potassium, and this is sodium. The potassium ions are more concentrated inside the axon than outside. So they will want to move out. When we look at this in a diagram from your textbook, it looks something like this. So we have these sodium ions on the outside, shown right here in green, and they are more concentrated than they are on the inside. So sodium is going to want to move in. We also have potassium ions more concentrated on the inside, so potassium is going to want to move out. Now, the inside is negatively charged, and there are these proteins that have this negative charge at rest that that cannot move. So they cannot move across the plasma membrane. So these ions cannot generally move. They can't move by themselves because of the polarity of the plasma membrane, but they move using protein channels. So if we look at these protein channels, we have some channels that are open all the time. So potassium is going to want to leak out and sodium is going to want to leak in. We actually have to expend energy to pump the potassium out and to pump the to pump the potassium in and the sodium out. We also have channels in this particular instance that are closed at some points and that open at others. So as that membrane potential changes, as the membrane potential changes as it goes down the axon, these voltage gated ion channels will open and they will close. And this allows for lots of movement of potassium and sodium into and out of the axon. So these ions are electrolytes, and you might think of like when you exercise and they say you need to replenish your electrolytes, and maybe you drink an energy drink like Gatorade that has lots of sugar, but it also has sodium and potassium and chloride. Those are ions, those are electrolytes that we need to have inside of our cells as well as outside of our cells in order for the proper functioning of the nervous system.